Hello, my name is Hayden Dickerson. This is my walkthrough of some of the research myself, Dr. Hanny Rivera, Carlos Tremonti, Leah Williams, Dr. Sarah Davies, as well as Dr. Thomas Gilmore conducted on the temperate coral Oculina arbuscula. Our goal here was to figure out how heat and starvation stress affected these organisms. Given the facultative relationship that these corals have with their algal symbionts, they can adapt a bit more readily to our rapidly changing climate, which is affecting the shallow water temperatures of their environment. This experiment looked at symbiotic arbuscula in order to expand upon previous studies that have shown how heterotrophy can mitigate temperature-induced bleaching. We hypothesized that increased food availability and an assumed increase in heterotrophy would mitigate bleaching and promote immune response. Shown here are the treatments and experimental design. Note that these colors match up with the colors of the figures in the results, and also that the figures in the results section line up with the labels shown here. Corals were initially put into seawater at 24 degrees Celsius. The heated treatments had their ambient temperature raised 1 degree per day and then were held at 33 degrees for 4 days at the end of experimentation, while the controlled temperatures remained at 24 degrees for the entire duration. We used PAM fluorimetry to measure photosynthetic yield and used that as a proxy for how well corals responded to thermal stress. The increased amount of food did not help maintain photosynthetic efficiency in symbionts subject to the heated treatments, with both showing a marked decline in FV/FM values after the onset of the heat ramp at time point 2. We also ran a ANOVA test to note differences in carbohydrate and protein levels across the 4 treatments. Neither chlorophyll concentrations nor most of the carbohydrate concentrations differed significantly from one another among the treatments, save for the decline in carbohydrate reserves of the symbionts in the fed heated condition relative to fed control. Protein levels were the most responsive to treatments, with corals showing a decline in protein concentration across all treatments relative to the fed control. Symbionts showed a similar relationship, however, the unfed heated samples were not statistically different from fed controls. The coral immune response was measured in levels of the protein nuclear factor kappa B, a transcription factor known to regulate innate immunity in animals. NF kappa B was strongly inducted in the fed heated treatments compared to the unfed control, suggesting that temperature is a stronger stressor than starvation in this organism. In conclusion, these results suggest that at short timescales, heterotrophy does not alleviate heat stress. Temperature instead is the dominant factor in both the immune response of the host and the photosynthetic efficiency of the symbiont. Despite the decrease in efficiency, chlorophyll concentration did not differ from one another significantly, while protein levels differed substantially between treatments. Determining the specific protein levels that were altered in response to the treatments could help further our understanding of how corals bleach. I'd like to take this time to bring attention to the references in the acknowledgement section. We'd really like to thank Hannah Eichelman, James Pfeiffer, Nicola Creefall, and Daniel Woodchick for feedback on data visualization and analysis, and generally being great people. They're all in the Davies lab, and it's a great place to be. Great team to work on. We also like to thank Brianne Dent and Justin Scase for aquarium and husbandry help, and lastly, Steve Broadhurst for sample collection.